We're going to be looking at protein synthesis part 3. This is on page 9B of your AP notes. Okay, so first of all, it talks about mutations, and mutations are caused by something called mutagens. And mutagens are something, they basically mean that they are able to generate a mutation. Okay, so there are several different things that can cause a mutation, things like UV radiation from the sun, cigarette smoke, excessive use of alcohol, viruses, even gases like car exhaust, chemicals, pesticides, insecticides, poisons. All those things can cause mutations to take place in the DNA. So there's two major kinds of mutations. One of them is called a point mutation, and the other is called a reading frame shift mutation. All right, so a point mutation means that it is just a single point gets changed. All right, so if this is our messenger RNA that we start with, and I would encourage you even to maybe write these down so that you can have something to compare and look at as you're talking about there or looking at those notes. A point mutation means a single nucleotide is mutated, okay? So in that, you're going to have things like a silent point mutation. All right, so if you look right here, all of these letters are the same on this messenger RNA. If this first one here is our normal, okay, this is our normal up here. Okay, what it's supposed to look like. All of those are the same until you get here and you have a U instead of a C. Okay, well that codes for GGC, codes for glycine. Well, even though a U was accidentally brought there instead of a C, GGU also codes for glycine. Remember, there are several amino acids that have multiple codes for the same amino acid. You've got 61 codes for amino acids and only 20 amino acids. So you've got lots of them that use more than one code. So in a silent mutation, there is actually no change because the transfer RNA is going to bring glycine either way, whether it is GCU or GGU. It is still bringing glycine. So the amino acid that's brought is unchanged. So you're going to get the same protein. So there is no change in the organism. You may never even know that has even occurred, okay, that it's happened. Okay, so then we have a missense mutation, point mutation. And this is where there has been a mistake, okay? And it changes the amino acid. Everything is the same except on this one point, you have an A instead of a G. That's supposed to be GGC, which codes for glycine. Well, instead, they brought in AGC, and it's just this one point that gets changed. But AGC codes for serine, so the transfer RNA brought in serine. Well, that's going to change the protein that is created. It's going to cause a problem there. Sickle cell disease, sickle cell anemia, if you remember from anatomy, where the red blood cells are sickle-shaped instead of circles, okay? This is going to be a cause of that disorder, okay? It's a missense point mutation. All right, so then the last one is a nonsense point mutation. Okay, you've got your AUG that codes for methionine, but right here, instead of bringing AG that is going to code for lysine, it brings in a U. Well, UAG is a stop codon. So it actually stops the process. And the RNA never really has an opportunity to create this protein. So this makes no sense. There's no protein, no protein, and it is a nonsense point mutation. Okay, then you have a frame shift a frame shift mutation, and it's called reading frame mutation. This is where everything gets messed up behind it. Okay, so this is our normal, what it's supposed to look like. All right, the first kind is that they're showing you here is that you're going to have a letter that gets deleted. So this is our deletion. Right here, you're supposed to have U on your normal messenger RNA. But instead, one of the U's got deleted. So it is going to shift everything down because remember we work in codons, groups of 3. So now you're going to have UUG. Well, UUG does not code for phenylalanine. It codes for leucine. So you wind up getting the wrong amino acid here, but because it shifts down, it affects every amino acid after that. So the only ones that are correct are these before the mutation occurs. Everything else gets shifted to the left one, and it's going to code for the wrong amino acids from there on out. All right, so then you also have the frame shift mutation where you have an insertion. It adds an extra letter, okay? So that's what's going on here. AUG is normal, then you add an extra U. So instead of AAG, it being the codon that codes for lysine, you have UAA, which is a stop codon, and everything after that is not going to matter because your stop codon is going to stop the entire process. Now, it is possible that it wouldn't be a stop codon, it would be another amino acid, but it is going to mess up every amino acid after that. Now, you can see this one, if you insert or delete 3 nucleotides, so the entire codon is missing, it's not necessarily going to be frame shifts. Everything's not going to shift to the left or shift to the right, but you are going to have an entire amino acid missing, which is going to mess up the protein that is being made. So just to give you an example, if you look at this, we look at it just in a sentence form, you can see if we use 3 letters for our words representing the codons, okay? If we insert just 1 extra letter, okay? All right, we're inserting an I there, okay? You can see it messes up everything after that, okay? It's not going to make any sense. That's an insertion, okay? In the same way, if we leave something out, say that we leave out our O this time, and everything shifts to the left, again, everything after the area that the mistake was in, right there, everything after that is messed up, okay? So mutations can happen several different ways, and those are in your notes. I hope this helped in understanding those.